Hello all. In this video we are going to see about paracrine signaling. Already we have seen paracrine signaling in previous videos with regard to stem cell niche and during morphogenic gradient. So, now we are going to see about how signaling transduction takes place with the help of sonic hedgehog. In case of developmental biology, we will be studying about anterior, posterior axis formation, dorsal ventral axis formation, proximal distal axis formation in invertebrates and invertebrata. So, for all these axis formation, we need signal transduction pathway. For the signal transduction pathway, these animals make use of WNT signaling, WNT beta-catenin signaling, and sonic hedgehog signaling. These two signaling mechanisms are very important with regard to the study of developmental biology. So, in developmental biology, we study about how a single cell form and develop into a whole organism. We make use of these two signaling pathway to learn about the axis formation pattern in developmental biology. In this video, we will discuss about hedgehog signaling. Next video, we will be discussing about WNT beta-catenin signaling pathway. Hedgehog signaling. Hedgehog act as a morphogen. So, we already know what is morphogen and morphogenic gradient. It becomes very easy to understand that hedgehog is a morphogen. First thing. Next thing, we will learn about types of hedgehog. Hedgehog can be divided into three different types. First thing is SHH, that is sonic hedgehog. IHH, Indian hedgehog. DHH, desert hedgehog. Sonic hedgehog, help in eye field specificity. Indian hedgehog, help in bone growth. Desert hedgehog, help in spermatogenesis. So, these three are the types of hedgehog. Now we will see about signaling pathway in hedgehog. For a signaling pathway to be studied, we should understand few things. First, we should know what is the receptor involved in the signaling system. We should know about receptor. Next, we should know what is the co-receptor involved. Third thing, we should know about ligand. Ligand is nothing but it can be a molecule or any other hormone or secondary messenger that comes to contact with the receptor. In case of hedgehog signaling, we have a receptor that is known as patched receptor. Receptor is patched. Co-receptor is smoothen. Ligand is hedgehog. We know receptor, we know co-receptor, and we also have appropriate ligand for this signaling system to be carried out. Now we will see the pathway. Pathway can be better studied by understanding in two different cases. First case will be studied with hedgehog absence. What will happen if the Heghog ligand is absent, and what will happen if the Heghog ligand is present. These are the two cases that we are considering with regard to Heghog signaling. First, we will draw a cell. A cell. Then we will draw ligand. We will draw receptor. Sorry, this is receptor. We know this receptor is patched. This receptor is patched. Okay. Then we will draw ubiquitous interrupters. This is the basic protein. Then we have molecules like protein kinase A, Kifi, Sufu, Slim. These are systems that inhibit ubiquitous interrupters. This will inhibit ubiquitous interrupters. So, protein kinase A, Kifi, Sufu will inhibit the activity of ubiquitous interrupters. Function of these three will inhibit the activity of ubiquitous interrupters. Then we will go and study what will happen if Heghog ligand is present, what will happen if Heghog ligand is absent. Before that, we will draw the co-receptor, that is smooth end. It will be present here. Name is smooth end. Will be present here. The name is smooth end. This is the name of co-receptor. We will see what will happen if Heghog is present. Heghog is the ligand. It comes and sits on the appropriate patched receptor. If it is present, then this smooth end will become activated. Once if the smooth end is activated, it will go and inhibit pKa. It will go and inhibit Sufu. It will go and inhibit Kifi. That is Kaifi. So these three things will be inhibited. If these things are inhibited, this ubiquitous interrupters will undergo transcription, translation, and it will form appropriate protein. And the function will take place. If the ligand is absent. If the ligand is absent, smooth end will not be activated. If smooth end is not activated, pKa, Kufu and another protein named COS2 and the protein named COS2 will continue inhibiting ubiquitous interrupters and no translation and protein formation will occur. Function will not occur. This is the major mechanism of Heghog signaling. Summing up in detail, if If Heghog ligand is present, smoothen gets activated. The activated smoothen will go and inhibit SUFU, it will go and inhibit pKa, it will go and inhibit COS2. All these things will be inhibited. So, Ci will happily go and undergo transcription and translation to form protein. Then, if the ligand is not present, smoothen will not be activated. If smoothen is not activated, pKa will continue inhibit Ci, SUFU will continue inhibit Ci, SLIM will continue inhibit Ci, COS2 will continue to inhibit Ci. So, transcription and translation of ubiquitous interrupters will not occur, and no protein formation occur, and no function occur. These are the basic signaling systems in developmental biology. Hope everybody understood sonic Heghog signaling pathway. This Heghog signaling pathway functions in various systems in case of digits formation and even in cancer biology as well. Hope this video is helpful to everyone. Thank you so much for watching this video. Thank you.